Welcome, welcome everyone to the closing plenary for ISEE Young.
So are we all in?
We are, I'm sure we're not all here, but we're 93, so I think we should go ahead and get started.
So first of all, in the name of the Scientific Organizing Committee and my dear co-chair Massimo, we want to welcome you to this last session of this meeting. We also wanted to thank very much all the local organizing committee. It has been a pleasure to work with you, and also all the technical part. And I think the conference has been very successful up to now, thanks to the work of everybody and also the presenters and also all the participants who asked questions. And even if it was not quite the typical conference we usually have, maybe we have to get used to these kind of things. And I am not sharing the right screen. I wanted to share this screen.
Please excuse the interruption, but someone has a phone notification that's going off. Maybe you can mute your phone. Thanks. Sorry, back to you.
Yeah, so I wanted to share this screen. That's, I think, if you're all looking at it, okay. So that's the kind of pictures we have from this meeting. Is not the kind of picture we usually have, but I think that was not too much of a problem to have a very successful. And we had a very nice yoga and Pilates session and very nice keynotes, buvette, rooms. It was almost as we were in person when we could have meetings, etc. So thank you very much to all of you. And now I give the room to Massimo.
Yes, thank you. Just to reinforce your message, a big thank you to everybody. Of course, to the Scientific Organizing Committee who shaped the program, of course, and to all the presenters and the attendees who made this conference so interactive and successful. And of course, a big thank to the local organizing committee, which made all of this possible. So a really big thank you from myself and Benedict. And I would now move to the first speaker of the session. It will be, as you will see, a very intense session full of presentations and full of people. So please stay with us until the end. I'm very glad to introduce the first speaker, Michelle Turner, Assistant Research Professor at ISEE Global and also Secretary and Treasurer of the ISEE. She will give a talk on the benefits of being an ISEE member. Please, Michelle.
Great, thank you very much, Massimo. I'm going to try and share my screen here. So I made just one slide for you today. So I'm the Secretary Treasurer of ISEE, and I'm responsible for kind of overseeing the finances of the society as well as the society membership. And ISEE is made up of about 1,000 members all around the world, and about 30% of the membership is made up of students and new researchers, like most of you here in the conference. So students and new researchers make up a very large proportion of the society. And so I have this one slide that shows the official membership benefits, which you may not realize all of the benefits available to you. And then I also have a few other comments I'd like to make, just offering my own personal perspective about participating in the society. So this is a listing of the membership benefits. Of course, you are eligible for things like reduced registration at the annual meeting, eligibility for conference awards, including travel awards or registration awards. There are monthly newsletters that are sent to all the members that advertise about different events and webinars and activities. There are professional development opportunities, and you can volunteer to participate in different committees, chapters, and different working groups. you are eligible for different career awards, not only at the all throughout your career, starting from awards aimed at student and new researchers, aimed all the way through the career, including Lifetime Achievement Awards. There are a variety of networking opportunities, including the annual meeting and other regional meetings and ISEE Young meetings like this one. There is a free online subscription to the journal Epidemiology and 20% off publishing in the journal Environmental Epidemiology. And if you're here from a low or middle income country, ISEE also sponsors publishing waivers in the journal Environmental Epidemiology. So you can request one of these waivers and we'll pay for your publishing fees for you to publish in that journal. You can also write commentaries to the journal Epidemiology, which are published at no cost to you, and also other discounts on books, other related society memberships, and also some new initiatives that are coming soon related to different online workshops and training courses, which are going to be developed more and more over the next few years, as well as eligibility for a new type of award called the ISEE Fellow designation that recognizes people who have contributed very actively to the society over an amount of time. And then, beyond these listed membership benefits, perhaps I'll stop sharing my screen now. When I was a PhD student myself, I was not a member of ISEE, and I didn't really know what, was it really worthwhile to join a scientific society, and was it worth paying, the membership fees? And I think that there are a lot of opportunities and advantages that might not be obvious at first glance, when you're looking at this list of membership benefits. And I think two main things come to my mind. One is that ISEE offers a great opportunity to participate and contribute to the research area in general. I think many people here are very interested to contribute more to research and to research organization in general, but don't know how to do so. And so you can volunteer to participate in all sorts of different ISEE committees or initiatives. We're launching some new initiatives this year related to communications, related to online training that I already mentioned, related to greening of the annual conference, and anti-racism, ethics and policy. And so these are all ways that you can get involved and make a big difference. If you want to get involved, you can contact the SNRN and they can connect you with the right committee or the right task chair. Most people here are part of the, who are living and working in Europe are also a member of the Europe chapter of ISEE. And you can also connect with them. I'm sure that they're always looking for volunteers to develop new initiatives. ISEE too is a very open and a very supportive and a very welcoming society. And so people, all of the committees and chapters and chairs and task groups, they're very happy and welcoming, very happy to welcome new members. And the second aspect about ISEE that I'm not sure if it comes through on that slide is the value of networking. And you have all been networking here over the past 2 days together online. And I think I want to stress to me that that's been a very valuable aspect of participating in ISEE. It can also lead to doing great research projects together internationally. It can lead to finding jobs. It can lead to helping you to grow your career in general. And also something that was mentioned yesterday by Barbara, it also can offer a lot of friendship and support as you navigate your research career. There's a lot of us, a lot of people that may be the only environmental epidemiologist at your institution. And so it's a great way to get connected and supported by others doing similar type of work. In the next few weeks, we're going to be posting soon the new strategic plan for ISEE on the website, the new strategic plan for the next 4 years. And so we will welcome all of your feedback and ideas and comments on the report. And if you have any new ideas on what you think ISEE should be doing in the next 4 years, please let us know. So I think that's all for me, and thank you for letting me present here today.
Thank you very much, Michelle. So I would say there are a lot of reasons why to register at ISEE. Okay, I think now we can move into our keynote session. I'm very glad to introduce our keynote session on microbiome and the urban environment. And for this, we have invited the two renowned scientists who have been working on this complex topic since many years now. They will help us understanding how our microbiome is influenced by the external environment and therefore how it can mediate the effects of toxic exposures on human health. Since the two presentations are on the same topic, we will have a joint discussion with questions and answers only at the end of the session. So I invite the audience to post their questions in the Q&A chat box, and we will select a few questions from the chat at the end. Thank you very much. So I'm happy to introduce now the first keynote speaker, Nina Iszatt, an environmental epidemiologist at the Norwegian Institute of Public Health. Her research focuses on the role of early life exposure to environmental chemicals in the development of adverse reproductive and child health outcomes, and the gut microbiome as both independent predictor and mediator. The title of her talk is The Microbiome, Where Environmental Pollutants Meet You. Please, Nina, the stage is yours.
Thank you, Massimo. I'd like to thank the organizers for inviting me to this very well organized conference indeed. I think you can see my presentation. Yeah.
Yes.
Okay. So I just move these. Okay, so thank you for inviting me to speak on the microbiome, where environmental pollutants meet you. So this is a can of Spam. It was found 5 kilometers below the sea in the Pacific Ocean. This picture was used in a news report to illustrate the extent of modern toxic pollution, so somewhere even as remote and inaccessible as the depths of the Mariana Trench. But yet another 5 kilometers below this can, at 10 kilometers deep, researchers found extraordinary levels of PCBs, banned chemicals. So these contaminant levels were considered extraordinary by the scientists because they were considerably higher than documented for nearby regions of heavy industrialization, meaning that these pollutants are pervasive across the globe, including to full ocean depth. So our microbes evolved with us well before the time of Spam in a can, and now and how they react and interact with environmental pollutants is still being understood. So today I'll share with you some perspectives on the microbiome and toxic chemicals in our environment. Bacterial cells are adapted to virtually every ecosystem on Earth. There are estimated 100 trillion cells in your body, and less than a third of these are human. So other 70 to 90% are bacterial and fungal. On the other hand, when it comes to unique genes in your body, 99% are bacterial, while about 1% is human. The magnitude of this is eloquently described by Dr. Martin Blaser, in his book, The Missing Microbes, which I recommend you read, where he says that humanity is just a speck in the massively bacterial world. We need to get used to that idea. We also need to get used to the idea that they play an important role in health and disease. Microbes are found all through the human body, from the skin to respiratory tract, and not least in the gut, where approximately 95% of microbes reside. The gut microbiota has both the largest number of bacteria and the greatest number of species compared with other body areas. So this complex community of microorganisms has co-evolved with humans through the millennia. The old friends hypothesis describes a mutualistic relation with humans whereby gut bacteria encode environmental signals, and that provides essential metabolic services for their host. But it's not all one way. For example, some components of breast milk, oligosaccharides, are not digestible by the infant directly, but only by bifidobacteria, which are crucial in the establishment of the infant gut microbiome. And fascinating recent evidence shows just how far back this relationship goes. Ancient fecal sediments were uncovered at the El Sorts site in Spain, where many Neanderthals lived some 40,000 years ago, and they provide the oldest fecal material discovered to date. And researchers identified dozens of bacteria found in the gut of both Neanderthals and modern humans, which suggests that these have played a major role in the advancement of humans for tens of thousands of years. Bifidobacteria are essential starter microbes, which also regulate immune defenses, were among the bacteria identified. So where does it all start? So this figure describes the development of the human gut microbiome. the establishment of the host microbiota symbiosis is driven both by developmental and environmental signals, especially early in life. Here, the infant gut starts near sterile and rapidly evolves and matures and increases in diversity to resemble an adult community at age 3. Prenatally, the nascent immune cells start with growth of the immune cells that will prepare the baby for exposure to the outside world. Then during birth, the infants receive their first dose of microbes from the mother, particularly during a vaginal delivery. A C-section baby, you probably heard their first gut microbes reflect skin microbes rather than vaginal microbes, and this sets the baby off in a specific type of gut development. So not only is birth mode crucial, but also gestational age in terms of how developed the newborn's immune system is. So this interplay between immune system and gut microbiome continues here at the mucosal barrier. And during postnatal colonization, the microbial signals influence the maturation of the immune system. Early life factors, particularly diet, so breastfeeding, formula, introduction of solid foods, further drives diversification of the gut microbiome. And other factors, including family size, antibiotics, hygiene, geography and living environment, all of these have changed dramatically in recent years. And research is slowly uncovering the extent of the role of the microbiome in acute and chronic disease later in life. So it's thought that this dramatic environmental and lifestyle changes the microbiome is now exposed to has caused an increase in modern plagues, the diseases involving immune and metabolic systems such as asthma and obesity and childhood diabetes. I just became aware of everyone up there, and maybe that's in the way, sorry. Not sure if that's on your screen or not. Stick that up there somewhere. Okay, so Dr. Martin Blazer and his student Laura Koch showed how disrupted microbiome during a critical development window could lead to obesity in mice. So the control mice were given or allowed a normal microbiota development, while the treated mice were given low-dose penicillin, which disrupted their microbiota and caused weight gain. So when the microbiota were transplanted to germ-free mice later, they then took on the obese phenotype. So this experiment established a proof of principle for the role of obesity, microbiota in development of obesity. We also went on to show that the composition of the microbiome in early life was associated with BMI in children in the Norwegian microbiota cohort, which was the first birth cohort established specifically to look at the microbiome. Here at 2 years, the infant gut microbiome explains 50% of the variation in the children's BMI at 12 years. Now, it could be argued that this could be confounded by similar diet or lifestyle that is already in play at 2 years. However, even at 120 days or 4 months, when the babies were exclusively breastfed, 20% of the variance in later BMI could be explained by the microbiome composition. These studies looked at the composition of the microbiome. However, it's not who's in there, it's what they do that really counts. An important role of the microbiota is to ferment dietary fiber into short-chain fatty acids, which then are absorbed by the host. Thus, the gut microbiome appears to function more like an endocrine organ. The short-chain fatty acids are important as they're shown to have a role in immunologic, metabolic disease, and neurologic diseases. And this figure tries to describe the pathways through which short-chain fatty acids can mediate microbiota gut-brain interactions. For example, through the immune pathway, the endocrine pathway, the vagal pathway, better known as the gut-brain axis, and also through other humoral pathways. So this bacterial metabolome that is produced in part by the microbiota interacts with the host metabolome, and they are influenced by various factors. So not just the microbial composition, but also environment, lifestyle, genes, and diet. And it's that interaction that can lead to phenotype and good health or disease. So where do environmental pollutants fit in? This is our exposure of interest at the moment. It can be that the chemicals are directly metabolized by the gut microbiome upon indigestion, as in A, or metabolized after conjugation by the liver, as shown here in B. In this way, they modulate toxicity for the host. Over 1,300 environmental core enzymatic families involved in the metabolism of more than 30 chemicals. In a second kind of interaction, the chemicals have a disruptive effect on the microbiome, inducing dysbiosis, so an imbalance in the composition of the microbiome, or they could interfere with their enzymatic activity of the microbiota. So although these interactions are described in experimental settings rather well, there are few studies in humans. We're particularly interested in environmental pollutants as they've been associated with diverse health outcomes, especially related to endocrine and metabolic systems, such as allergic disease, obesity, and neurodevelopment. And these are similar to some of the diseases now associated with the gut microbiome. So we therefore want to know if the microbiome is one mediator of the toxic action of environmental pollutants, in particular during this crucial developmental window that happens in the first 1,000 days of life. So we looked at this also in the NOMIC Birth Cohort. Here we looked at 28 persistent organic pollutants that were measured in breast milk and sampled at 30 days postpartum, while the microbial composition and short-chain fatty acids were measured in their infants' fecal samples. So 1-brominated flame retardant and PFOS, perfluorinated compound you've probably heard of, were associated with less diversity within individuals, alpha diversity, but these chemicals accounted for just 4% of variance in the diversity. So not hugely strong effects, not hugely influential. On the other hand, more chemicals were associated with disruptions to the short-chain fatty acids. So shown here for acetic acid and propionic acid. It's all the chemicals that we tested, and it's the blue results that we're particularly interested in here. This is the result of a multi-pollutant model. And so in general, the chemicals were associated with 25% decrease in short-chain fatty acids. But of note, PFOA, which is best known as a water contaminant, and also one of the dioxin-like PCBs, were actually associated with a 50% increase in short-chain fatty acids. But altogether, the chemicals explained around 30% of variance in these short-chain fatty acids. So this suggests that in this instance, at least, the influence of persistent organic pollutants is less on the composition and more on what the bacteria are doing. However, in an observational setting, it's quite difficult to interpret exactly what is happening. Are we seeing a direct toxic attack on the microbiome? Or is it that the toxic chemicals that the baby was exposed to prenatally and that are also highly correlated with the breast milk levels in the mother, are these affecting the baby's physiology and immune functioning, which then leads to change in the gut ecology? So it's that interaction between the immune system and the development of the gut. But it could also be that these bacteria are able to metabolize chemicals and they have a competitive advantage. So more of them and then less of other bacteria. So in sum, it could be any or all of these effects, and it's a bit difficult to untangle in this way. On the other hand, the role of the gut microbiome as a detoxification route for heavy metals is much clearer. For example, it's been shown, at least experimentally often, it's been shown that bacteria in the gut can metabolize arsenic and influence methylation, bioavailability, and excretion. So we also looked at this in the NOMIC Birth Cohort. So mothers' microbiota from fecal samples that were collected at 4 days were later associated with arsenic that was measured in her breast milk that was sampled at 30 days. So here you can see as the mother's diversity increases along the x-axis, here the alpha diversity of her gut microbiome, as the gut diversity increase, the arsenic levels decrease. This is shown for total arsenic, and it also was seen for DMA, which is one of the species of arsenic. So there was also a differential abundance of certain genera between high and low organic arsenic, which suggests they play an important role in the internal dose of the contaminants and their metabolites. This points to the potential of the microbiome as a target for intervention. So this is exactly what was tested in a randomized pilot study in Mwanza in Tanzania, where the population is highly exposed to arsenic, mercury, cadmium and lead through the consumption of fish. In this small but compelling trial, including 60 women and 44 children, the intervention group was given a yogurt containing Lactobacillus rhamnosus, while the control groups had whole milk or no intervention. So the intervention was protective against increases in mercury and lead blood levels in the pregnant women. So where to go from here? We're still lacking a basic understanding of the role of a wide range of pollutants on the microbiome, both in terms of its composition, but particularly its function, and also its mediating role in health. And in observational studies, this does require large cohorts with long follow-ups. But from this basis, it will then also be possible to investigate interactions between complex mixtures and exposome and both the bacterial and human metabolome. And finally, future public health interventions to reduce chemical exposure and improve health. There's the exciting implication of this kind of research. Whether this means probiotics delivering one bacterium, as was seen in the Tanzania trial, or if it's a controversial poo in a pill, which is hotly debated, or whether it's a whole microbiome transplant, this is very unclear. A third possibility is green prescriptions to replace what we've lost moving from nature to the cities. But this is the topic of the next keynote, and I will leave this to Virissa Lenters to describe. Thank you. I would also just like to acknowledge my co-workers, and in particular Mariana, who established the humus NOMIC Birth Cohorts nearly 20 years ago.
Thank you very much. And so as Massimo said, we will go straight to the next talk, and then we'll have a question and answer session for both at the end. So I am very happy to introduce you Virissa Lenters, who is an assistant professor in environmental epidemiology and exposome science at the University Medical Center in Utrecht in the Netherlands. And she currently leads research on how the urban and chemical exposome affect child development and disease susceptibility, including the dependent and mediating effects of the microbiome. So thank you very much, and the floor is yours.
Thank you. Give me 1 second, please, and I'll share my slide. Is this, you can see my presentation, yeah?
Perfect.
Great. So thank you, Nina, for that nice introduction. And I'm going to talk about some of the aspects of our green environment, our city environment, and how that's influencing our microbiome. So we know still relatively little about the environmental predictors of the gut microbiome, and Nina already covered why the gut microbiome is so important for health and for our risk of many diseases. We know some things from farm studies, the Hutterites, for instance, are important studies in Europe, Gabriel, which many of us have worked on, that have given us hints at how our environment can be so important for not only allergic diseases, but also diversity of the gut microbiome. But those early studies still, gave us a small little snapshot into the influence of urbanization on our microbiome. So we're becoming more urbanized. More than half the world's population is urban, and that will only increase. And this means that this comes with all sorts of changes in our diet, in our housing, how much time we spend outside, what kind of households we live in, and also our exposures to all sorts of chemicals and pollution and medication. So this raises the question, if this pace of urbanization is outpacing our coevolution with the gut microbiome, are we maladapted to live in cities, or specifically is our gut microbiome maladapted for these modern cities? So I'm going to share some early evidence, emerging quite exploratory evidence. One of these landmark studies was by a group at Stanford, and they studied Hadza hunter-gatherers, one of the remaining hunter-gatherers societies. And they compared this with populations from 18 different countries. And what they found was they could really distinguish, this is a principal coordinates analysis where we're looking at the dissimilarity of the gut microbiome between groups. They could really distinguish the high income countries from the low middle income countries. and the Hadza are the sort of green color up in this top box plot, and the second axis is age. So those are the two most important factors, geography and age, that discriminate these microbiomes across the world. Other things that they found: a decrease in bacterial diversity in more modern or high-income countries. They saw disappearance of some taxa, and this has really raised some alarm bells in the community. And they also saw something where there's a trade-off between certain species of bacteria that are more prevalent in the Hadza and less prevalent in industrial communities. And this is one notable example where both of these are part of the Bacteroidetes phylum, and the question is, are these replacing the functional roles of those, or are we losing some really crucial functions? So another similar study carried out by Rutgers University, they studied 4 locations across the Amazon River in South America, and this was along an urbanization gradient, so a village in a jungle, a town, more rural community to the city, and then they divided the participants into lower and higher socioeconomic position within that city. And they observed big changes in the building materials and the housing, not surprisingly. So all sorts of different materials being used to build those homes. They sampled various locations in the homes across the body and different microbiome sites. And they found that the chemical diversity increased in those homes. So the city homes had many more types of chemicals in those homes, notably a lot of cleaning products. And they also noticed a decrease in microbial environmental exposures, what people are exposed to outside. And they saw that there were quite some associations. This is a correlation analysis, but quite some associations between different bacterial taxa or microbiome and various chemicals, notably here, cleaning products. So this one is in our shampoos, on the top right of this correlation analysis. They also saw a little bit against what they expected, an increase in fungal diversity in the houses. So there's more cleaning products, less bacterial species, but an increase in fungal diversity in the houses and on the skin microbiome. And then, as expected along lines with earlier research, there was a decrease in gut microbiome diversity with the increasing urbanization gradient. So research that I've been working on with colleagues at the University of Groningen, or University Medical Center Groningen. We're trying to look at a broader set of factors, also factors that we label exposome in our fields, and associate that, see how that influences the gut microbiome, not only the composition, but also the function, antibiotic resistance genes, virulence factors. And to do this, we looked at a very large population of 8,000 individuals. It's a multi-generational cohort, and they're spread throughout the Netherlands, but more concentrated in the northern 3 provinces. And we also looked at 241 factors, things, anthropometrics, medications, diet, health and disease status, different indicators, and also some exposome factors or environmental epi factors, such as air pollution, which I'll get to. So first I want to emphasize that our gut microbiomes are very individual, and they're also individual not only in their composition, so each of these little lines represents a single participant in the cohort, but also in their function and in their resilience, how quickly they rebound from a stressor. And we found, this is confirmed by many studies, but in this large study, we confirmed that the environment is much more important than genetics in shaping our gut microbiome. So you inherit some taxa from your parents, that genetics do determine in some ways what the composition of your adult microbiome looks like, but the environment is much more crucial in shaping what your gut microbiome will look like. But as I mentioned, we still know very little about what that environment, that 80% environment means. In this study, we found that living with somebody was very important. So this is comparing siblings that live together on the left-hand side, the pinkish box plot, with siblings that have been separated, the green one, compared to a random pair of unrelated people. And we see that once you move out of the home, your microbiome becomes just as different from your brother or sister as it is to a random stranger that you would see on the street. So we know very little of it is about those factors in the home that are shaping that. We've had only some very small studies that have tried to characterize that to date. We looked at several aspects of also our outdoor environment, air pollutants, green space or biodiversity markers, population density, and the socioeconomic position of your neighborhood. And one I wanted to highlight as population density is associated with alpha diversity. So some early studies showed that the farmers in different communities tended to have higher alpha diversity. In this study in the Netherlands, I should give a big disclaimer that the Netherlands is a very unique country with its dense cities and its highly industrialized farming. But here we found that urban had richer, more diverse gut microbiomes than rural communities, and rural representing largely industrial scale farming communities. And when we narrowed down to specific species, there are many different bacterial species and taxa that are associated, but some of these are also associated with health outcomes that we've established clear relationships with health outcomes. So I'm just going to highlight a couple of results from this analysis. First, I want to say that we still, with this rather quite highly powered study compared to previous ones, we still only explained 13% of the variance in taxa or the composition of the bacteria in our gut, and 16% of the functional profile. So that's relatively modest. But interestingly, these exposome factors, urban exposome factors, and also things about our indoor environment, such as pet ownership, explain quite a large proportion if we compare it with some other more traditional risk or factors, such as diet and medication use. So after adjusting for some technical factors, then we have more than 4,000 associations between all these 240 factors in our taxa. And we find that the more established ones like age, sex, BMI, the Bristol stool scale characterizing what your poo looks like, and the season fecal sampling are most important. Those explain the largest proportion of variance. But we also discovered that some exposome factors, both in childhood and current exposures, were very influential. So urbanicity, so how, whether you live in a city or a rural environment, whether your parents smoked in childhood, those were associated with your adult microbiome. These associations were stronger than those observed for birth mode, whether it was a cesarean or vaginal birth, for breastfeeding, and for preterm birth. So just to reiterate, this is things that happened to the baby. These associations can be observed in adulthood, but these associations are stronger than the more traditional ones that we see that influence the infant developing microbiome. And then current exposures, pet ownership in the home, definitely affecting the microbiome of the home. And also interesting, green space outside of our home affect our composition. And air pollutants were also associated, then rather in the opposite direction, and some of these associations were also associated with health outcomes. So this is where we're doing a mediation analysis. This is very much a new field. I know earlier today, Léa Maître presented some really exciting research, also looking at exposome factors in relation to the child microbiome. A lot still needs to be done in this field. And one thing that we really need to work on in the exposure science field is addressing how to do better exposure assessment. So I was talking about the degree of variance explained, but that's maybe not quite fair because we have a lot of different measurement error, levels of measurement error on these different factors, and we can't compare them, or we need to carry that caveat and do better jobs of weighting that error. We also know that it's very dynamic, not only our exposures, but also our gut microbiota. So some of our genera, for instance, show distinct seasonal patterns also in modern societies. So we need to account for this, and we've done a rather simplistic job of doing that so far in the field. We also need to use more untargeted approaches, internal exposome approaches like high resolution mass spectrometry, to look at things like, well, people have hypothesized food preservatives, microplastics are very important, but we still have very little data on this. the implications for health. So we're learning more and more how the microbiome is important for immunological, allergic diseases, but also mental health, cancer, all sorts of diseases. But it's important that we pair these association studies with more functional studies, and that's happening more and more. And to apply some causal approaches when we do our epidemiology studies. So is the dysbiosis or the sort of shifts we observe in the gut microbiome, is that a causative agent or is it a consequence of the disease itself? The classic correlation versus causation debate. And then lastly, and Nina touched on this brilliantly, it would be really exciting if we can have interventions and modifications to our gut microbiome. So these are at the policy planning level. Can we design healthy urban settings that promote a healthy gut microbiome and microbiome on other sites like our skin and our respiratory microbiome? Can we create home environments with respect to the cleaning products we use and all sorts of chemicals? And can we also modify our diet with respect to micronutrients like selenium or others, and prebiotics that feed the good bacteria to optimize our microbiome and also make us more resilient to the inevitable pollution that we're exposed to? So these are things I'm excited about, and I look forward to discussing with you in the question and answer period.
Thank you very much. That was really inspiring. Congratulations to both you and Nina for these presentations. So this is now a good moment for people to pose their questions in the chat. I don't see questions so far, so I invite the audience not to be shy and to pose your questions in the chat. Meanwhile, I will start by admitting I'm not an expert in this topic. So I'm sorry if my questions might be trivial, but what I can understand from your both presentations is the very complexity and the high dimensional data that you are handling. I mean, I see a lot of potential exposures, a lot of potential, effects, intermediate effects, and ultimately some, let's say, visible health effects on the subject. So is there a way to kind of eliciting this causal chain? I mean, how do you kind of try to simplify all this huge chain of events in order to better grasp what's going on? I mean, do you have any suggestions, since this is an ISEE Young Conference, suggestion for young investigators willing to follow your steps? I mean,
I don't know if simplifying is, I think we need to work in multidisciplinary teams, so that we're using better statistics. Some of the statistics are rather primitive from an epi sense. When we look at beta diversity, we're using tools that don't adjust for confounders properly, perhaps, or we can't adjust for correlated co-exposures very easily in some of these statistical approaches. So I think we need to rather improve the complexity of our modeling, but also pair that with some functional studies, as I mentioned. So where we do a mouse experiment and we expose the mouse to air pollution, there's some recent small studies that have just started to do that. And I think we need those studies too to address some of these questions about, what are we actually seeing?
I also have a question, a very, very basic question. So first of all, I'm sorry because it's very naive, but what is the difference between microbiome and microbiota?
Yeah, sorry, maybe I'll let Nina jump in.
Yeah, so the microbiota is the bacteria and also the virus and the fungi, and then the microbiome is the whole of them together. So we talk about the mycobacteria mostly for the microbiota, mostly for the bacteria.
Okay.
And then the microbiome is the whole, as a whole, the ecosystem.
Okay. Also, yes. And what is the relation between the different microbiome, the gut, the skin, and in the same person? Are they highly correlated or not?
So we don't know. Well, we have some very interesting studies about how the mother's skin bacteria affect the child's seeding and their development of their gut microbiome through breastfeeding. But we still need to learn a lot more about that. So there's big questions about the gut or the and lung axis, how the respiratory microbiome and the gut microbiome are interacting. And that's still a rather big question mark. So we study them generally as distinct microbiomes. But the crosstalk is, and I should say also the gut microbiome is huge in terms of abundance and weight and how much is in there versus your respiratory microbiome. There's also some debate in the literature whether there is a placental microbiome or how sterile our breast milk is. So I think a lot of these questions still need to be answered. And Nina, please add to this.
No.
Also, in your study, if I understood well, you will find a greater diversity of microbiome in urban than in farmers in the Netherlands. It's in the opposite way of what you have expected, I guess.
Yes, it is.
Okay.
Yeah, and we can't explain this yet. So this is definitely where we need to dig further into the data. And I think this would be unique to the Dutch situation. So there's been some really important work done by Rob Knight at the University of San Diego, where they've the Human Microbiome Project, they've characterized populations across the globe. But we still, it's still rather on broad lines, where we look at high income versus low income, or a diet very heavy in certain types of fiber versus not fibers. And so the more subtle nuances that are shaping our microbiome and sort of across Europe, for instance, are really,
But what about the role of environmental antibiotics?
Yeah, so colleagues of mine at Utrecht University are very interested in studying this, how this is, how in the Netherlands we've reduced a lot of the antibiotic usage in livestock farming, and what kind of influence that's having on human microbiomes. And that, I think that's still too tentative to say definitively, but I'm not going to address that, but people are looking into this. I don't know the answer.
Thank you.
Okay, so there are a few questions from the audience now. So the first for Virissa, could you give an example of how urban planning could promote microbiome? You talked about population density. Are there any other urban factors?
Yeah, so I think harsh chemicals are unfavorable to healthy microbiome. So from hospitals, we know that if you leave the window open or you clean using friendlier cleaning products, that we have a healthier microbiome. There's always going to be bacteria everywhere. So just trying to kill it isn't going to mean that there's none there. So it's better to have more natural ways of dealing with our microbiomes that are inevitably in our homes and outside. So I think the same could go for our outdoor planning. You know, there's a lot of efforts to increase green space for our well-being, but that green space is also going to increase our biodiversity or diversity of microbes that we're exposed to in the air. There's been a recent study showing that soil diversity of microbes has a positive influence on allergic disease development in mice. So that would be some first ideas I would have is, greener spaces and careful with our cleaning of the outdoor spaces.
Okay.
In relation to the greener spaces, it's not just any green space. Obviously, you know, if it's this manufactured green spaces where you've only got one kind of plant or something, is obviously, and you need to use a lot of chemical input to maintain it, there's obviously going to produce a much lower diversity of bacteria to be exposed to, compared to something that's much more diverse and natural, like a forest, or, you know, something which isn't maintained in quite the same way by humans. So it's also the kind of green space that
Thank you, Nina, because to elaborate about this, because there was a question from Mark, exactly how much green space exposure do we need for a good microbiome? I think you just answered that it's also the quality and the diversity of the green space which matters. There are other questions, of course. One quite general. I believe we are just tapping the knowledge to be discovered in this area. However, is there some general advice to everyday life that can be given, supported by evidence, like eat more yogurt or fermented food, eat more fiber, take probiotics, this kind of advice?
From what I understand, we have a hard time shifting our gut microbiome with dietary changes unless you go through a really profound dietary change. So, drinking a little yogurt drink, we don't have the strong evidence that that's going to lead to improved health. So there's a lot of companies trying to sell this, but that's still too early, I think, to try to be doing those things. I think early life interventions, there's some robust evidence for that. So C-section versus vaginal birth, that can have an influence on our gut microbiome, but what that means for adult health, I think we're still uncovering. And repeated antibiotics, that has been shown to really have dramatic negative influence, profoundly shift your microbiome. So you don't want to be giving repeated antibiotics if you don't have to, definitely not to young children.
But the interesting thing is that like one dose of antibiotics, your microbiome can recover. So given the chance, it can recover and it will return to its former state, but it's the repeated that causes a problem. One sort of anecdotal possible way of changing your microbiome was just before the pandemic, I was at a dinner with Rob Knight, who's one of the pioneers in this field, especially computational biology. And along with some other microbiome researchers, they were very keen to share a glass of wine because it was that you could share the bacteria, but I'm not so sure that this is something anyone would advise now in days of COVID. So yeah, depends a bit on your perspective.
Thank you. There are several questions. I will just pick some. This is for Nina. Regarding the treatment of dysbiosis, what data are you telling us? Is an intensive treatment with probiotics probably more effective versus fecal transplantation?
Yeah, I'm not sure that's known. I think the thing with the fecal transplantation is that there's a big concern, although it's could be helpful with some infections. There's a concern that you don't know what else you're transplanting. So what other diseases or what's the microbiome also going to contribute to in terms of health and disease? It's not very well known. So I think it's quite a controversial thing. So you might stick with the probiotics, but I think even with the probiotics, it's still quite sketchy evidence.
Interestingly, there is another question related with the COVID crisis that you just raised a few minutes before. Are you expecting changes in the microbiome with all the hand washing and disinfection going on with the COVID-19 crisis?
Yeah, I think absolutely, and especially in the younger, in the children that are the babies, the infants, and the early age school children who are doing incredible amounts of hand washing. One of the first things was this idea of, I don't think either of us touched on it, but this hygiene hypothesis that it's as we're getting more hygienic, so we're losing bacterial diversity. And so we're really returning to that. And maybe you could expect to see an increase in asthma and allergy and that sort of thing following this. But yeah, it's a very difficult trade-off. Yes,
of course. And for Virissa, based on your previous answer, would you think diet should be personalized for people based on their microbiome, which could help to prevent non-communicable diseases incidence?
I think, the precision medicine that we're moving in the direction is very exciting. I think we're still a ways off from that. But yeah, I think that that could be promising, especially because genes we have limited ability to change those, CRISPR aside. But with microbiome, we know that we should be able to shift. We haven't found very effective ways to do it, except for dramatic shifts with antibiotics and diet. But we should be able to shift it. It's modifiable, unlike a lot of other omics that we're dealing with. So I think it's promising, but and we're still far from that, being able to know what diet you can have to have. I think there's some obvious things like fiber, unprocessed fiber feeds short-chain fatty acid development, that your gut microbiota can make those, and those have been associated, a bunch of those have been associated with positive health outcomes. So a healthy Mediterranean diet or high fiber diet and vegetables and fruits, and that has been shown to be good. But beyond those sort of broad sweeping statements, we don't have any magic species or magic superfoods that are really super promising. People are excited about kombucha and a lot of these drinks, but I think it's still a little bit premature.
Okay. Going back to your data, there is a question about this that maybe you can help us interpret it. Have you investigated whether the differences in microbial diversity between urban and rural Dutch participants is primarily attributable to dietary differences? So,
yeah, we tried to adjust for this, but in yeah, that didn't remove all of the effect. So this is the challenge we have, is indeed trying to disentangle.
Yeah.
A lot of these urban factors are highly correlated when it comes to air pollution and diet also. So that didn't make the effect disappear, but we need to explore this further. And I should also say that the dietary assessments are rather crude. It's food frequency questionnaires. We haven't looked at metabolites, for instance, from a metabolome analysis. So it did remove part of the effect, but not all of it. But I think we need to look into this in greater detail.
Virissa, could it just be like agricultural pesticides?
Yeah, I think that could be really interesting if we could characterize that more fully, because yeah, Netherlands is quite unique. It'd be really fascinating to do this in Norway, where we have forests. So that could definitely, yeah, be playing a big role. Yeah.
Thank you. I do not see other questions from the audience. I don't know, Benedict, if you have any further.
Well, this kind, is there any evidence about the organic food versus non-organic food on the microbiome?
Yeah. Nina, do you want to help me out here? I think there's some very small studies, but with the microbiome, yeah.
Okay.
I don't think there's good quality studies that have done, that are bigger than a few people that have good quality exposure assessment that have shown that. That would be I think food preservatives is one where they've done some experimental studies in mice that have shown that food preservatives are definitely very influential. But I'm not aware of organic versus non-organic.
But you would expect that, you know, the less treated, more likely to have diverse bacteria on, then that will be transferred to the gut. So.
Yeah, thank you. No, I don't see any more questions either. Someone? Okay. Well, it's just in time, I guess. Thank you very much. It was very interesting. Still a lot of research to do in that field. Very exciting. Thank you.
Thank you very much.
So maybe now we will have a short break, 10 minutes break. So the speed talker can prepare, and you can stay here, turn off your camera, have a real coffee, something, and we'll be back in 10 minutes at 4:15. So we are very happy to start this session of Speed Talks. And as you all know, we had one Speed Talk winner for each Speed Talk session. And now the 6 of them will present, and at the end we will all vote. So don't forget to vote at the end. So we'll start with Alexandra Burgler from the Swiss TPH, and she is here already.
Yes, I'm here.
Okay.
Hello.
So whenever you want.
Thank you. Can you hear me well? Can I just check?
Good.
Perfect. Okay. So imagine that it is a beautiful spring day and Millie is going to have her first date with David, her new crush. But Millie has had a sleepless night, finds it hard to breathe, and also has trouble focusing on her tasks today. But this is not because of David. It's pollen season. And Millie is not the only one who is affected by that. Actually, every 5th Swiss person is suffering from a pollen allergy. And while pollen allergies can elicit the typical symptoms such as itchy eyes and sneezing, potential more systemic health effects like on the heart, the brain, and the lungs are poorly researched thus far. At the same time, there's also a trend that pollen is released earlier in the year and in larger amounts due to advancing climate change. Thus, it is critical that we investigate the effect of pollen concentration in the air on our health. So we designed the epochal study, who is going to investigate exactly that in allergic and non-allergic participants. We will investigate the effect of pollen concentration on these 6 outcomes: on heart rate variability and blood pressure, lung function and airway inflammation, cognitive performance, allergic symptoms, sleep, as well as overall well-being and quality of life. We are going to follow our participants over a 6-week period at 16 time points using portable health technologies. We aim at finding those response relationships between pollen concentration and these health outcomes. And furthermore, we are interested in particular plant allergies. Do they have a differential effect on these health outcomes, or also higher sensitizations towards these pollen species? Furthermore, we want to investigate if there are particular vulnerable subgroups in our population, for example, the asthmatic group. So, what do you think is Millie going to do now? Postpone her date with David. As she has luckily read in a news article recently published by Alexandra Bögler and her colleagues, she is now well aware that her symptoms such as tiredness and difficulty breathing and focusing could all be well related to the high pollen concentration today. So she decides to call up David and suggests to meet him around the block in a new bar that has installed pollen filters. The ethical study will advance our understanding on the pollen effects on health, as well as inform policy making and contribute to individual forecasts of symptoms in the future. Thank you very much for listening.
Thank you very much.
Oh, stop share here. Great.
Good. So there will be no questions. Now we'll go straight away to the next speaker, Sarah Lucht. So Sarah is from the Heinrich Heine University in Düsseldorf. And
okay, can you see my screen?
Now we see you and your screen.
Okay.
So whenever you are ready. Thank you.
Hi, my name is Sarah Lucht, and today I'll be presenting work from the 1000 Brain Study. This is a cohort study in Germany on aging that took place between 2011 and 2015 in the Ruhr area of Germany, here highlighted in the orange on the map. And there were 578 participants whose data I'll be presenting today. They were on average 67 years old, and they had their MRIs, and they were evenly split between women and men. We looked at 5 long-term air pollution exposures, PM10, PM2.5, PM2.5 absorbance for soot, and then a quasi-ultrafine particle exposure here for particle number concentration in the accumulation mode, PNAM, as well as nitrogen dioxide. We also looked at 2 long-term noise exposures, 24-hour noise, as well as nighttime noise. Participants were asked to lay in the MRI, to close their eyes, but to not fall asleep. And this is because we were interested in the default mode network of the brain. This is a specific network involved in resting state, so it's known to be active during daydreaming, but inactivated during task activities. And you can see the 3 regions here. There's the lateral posterior DMN, which is in the back on the outside, the medial posterior DMN, which is in the back on the inside, and then the anterior DMN at the front. And we looked at structural parameters here. So we looked at 2. First is cortical thickness, which is highlighted in the green arrow. It's just the thickness of the gray matter in the cortex. And then the second measure we looked at was gyrification, which is a measure of cortical folding. So how curvy is your brain? And so you calculate that by getting the surface area of the really curvy brain down here and dividing it by the smoothed outer surface to get sort of these maps. And both higher gyrification as well as higher cortical thickness are healthier or better, you could say. And so the question I'm interested in answering is, do we see effect modification by sex on the associations between environmental exposures and brain structures in this work? The answer is not really. So these are the results for the gyrification index, and you can see it split out by left and right hemispheres, and then the 3 regions. Men are in the green, women in the blue, and then we have the different exposures at the bottom. They all look very similar here for the gyrification. The only exception might be here in the lateral PDMN, where women seem to have a positive association compared to men on both the left and the right. For cortical thickness, again, we see very similar results across men and women, with again, a few differences slightly here in the front. So the ADMN, we see inverse associations primarily limited only to men, and the reverse in the lateral PDMN, where they're limited almost only to women. But the long story short is that we saw no strong effect differences between males and females, at least in terms of air pollution and noise on these structural parameters in this network. Thanks.
Thank you, very nice. Okay. Now we'll hear a Yikun Man from the Yale School of Public Health in the US.
Hey,
Hello.
can you see my screen? Hello.
Okay.
Hey.
Whenever you are ready.
Okay. Hello, everyone. COVID-19 deeply influences our lives, and that's why we are not meeting in the beautiful Basel now, but in Zoom instead. Given the seasonality of influenza and infections from other coronaviruses, people were wondering whether the transmission of SARS-CoV-2 is also influenced by meteorological conditions. Here, our group aims to estimate the relationship between air temperature or specific humidity and SARS-CoV-2 transmission in over 900 US counties from March 15th to the end of August in 2020. Note that specific humidity is just one of the measurements of humidity, and in our study, we estimated daily reproduction number RT to represent the transmission of SARS-CoV-2 in each county. As indicated by the figures on the left, county-level data were estimated or collected for the analysis. For the mean model, we applied a distributed lag nonlinear model combined with generalized additive mixed models. We adjusted for many county-level covariates, such as population density and old population. We also controlled for the spatial temporal variations in RT brought by policies and other unmeasured confoundings using flexible smoothing splines. Here are some preliminary findings. From the curves below, we can see both lower temperature and lower specific humidity were significantly associated with increased reproduction number. We then calculated the fraction of RT attributable to temperature and specific humidity. About 5% of RT was attributable to temperature, and about 15% was attributable to specific humidity. When we further investigate the distribution of these attributable fractions AF, we found that temperature and SH contributed more to the transmission in colder and drier counties and months than in warmer and more humid counties and months. This study indicates that dry and cold weather are moderately associated with increased transmissibility of SARS-CoV-2, with humidity playing a larger role than temperature. And since it's already 2021 now, we are currently updating this study using extended period of data. Thanks for listening.
Thank you.
Thank you very much. Our next speaker is Carly Goodman from the York University in Canada. Please, Carly, whenever you are ready.
Hello. Thank you very much for the introduction. So neurodevelopmental disorders are on the rise. From 1997 to 2017, the prevalence of having any one neurodevelopmental disorder increased from approximately 13% to 18%, with especially increasing rates among boys. Evidence shows that exposure to neurotoxicants such as lead and mercury can be a contributing factor to neurodevelopmental disorders. And interestingly, in the literature, we see sex-specific effects from neurotoxicant exposure. So the purpose of our present study was to determine whether boys or girls are more vulnerable to IQ deficits from early life exposure to neurotoxicants. We were specifically interested in 7 neurotoxicants known to impact IQ, namely lead, mercury, polychlorobiphenyls, polybromide diphenyl ethers, organophosphate pesticides, phthalates, and air pollutants. We were interested in exposure during the prenatal and early life period, as that is when the brain is most susceptible to neurotoxicants. We screened abstracts from 1950 to 2019 and found that out of 101 studies that tested for IQ, 33 tested for sex effects. When we look at how this has changed over time in the literature, we can see that a greater proportion of studies tested for sex effects after 2005. This seems to be in direct response to a workshop held in 2005 calling for all studies in toxicology to assess for sex differences, indicating that this is an important area of research that has previously been overlooked. Out of the 33 studies, we can see that 9 found differences between the sexes. And if we look at the pattern of results, in the prenatal period, 4 of 6 studies found an effect of lower IQ only in boys. In contrast, though, in the postnatal period, 2 of 3 studies found an effect of lower IQ only in girls. This suggests the potential for sex-specific vulnerabilities across windows of exposure, albeit with stronger evidence in the prenatal period, which might be due to the neuroprotective properties of the female sex hormone estradiol. But why should you care about this? Well, these subtle decreases in IQ can make a big impact on the population level. When you look at a normal distribution of IQ scores, approximately 6 million children will be considered challenged and 6 million gifted. But when you look at a lead-exposed population, the curve shifts to the left, effectively increasing the number of children who are considered challenged and decreasing the number of children who are considered gifted. So if we can identify who is most vulnerable to these neurotoxicants, then we can help with policy changes to prevent the shift of this curve. This study should be a reminder for the importance of testing for sex-specific effects in neurotoxicology. Thank you very much.
Thank you very much. Next, we have Annalisa Sheehan from Imperial College London, UK.
Hi, 1 second, let me just find the button to share my screen. Brilliant. Okay. Hi, so yes, I'm Annalisa Sheehan and I'll be talking to you about my PhD project on the
Annalisa, we're not seeing your screen.
Oh, okay, sorry. Let me try that again. Can you see it now?
Gorgeous, there it is, yeah.
Okay, brilliant. Okay. Great, okay. So yes, I'll be talking to you about my PhD project on the geospatial estimation of non-exhaust emissions using remote Earth observation. So emission inventories are key in determining significant sources of air pollution and provide vital data used in air pollution modelling. So emission inventories are constructed by multiplying activity data and emission factors. So activity data includes, for example, kilometres driven, population, vehicle count and road length data. And emission factors include grams per kilometre driven. And for this talk, I'm going to focus on vehicle count. So vehicle count data in cities are often based on manual counts or automatic number plate recognition data. And a limitation of these is that they are expensive, are only at point locations, and often only capture a snapshot in time, so are missing vital information on spatial variation, annual trends, and traffic growth. So for my project, I'm aiming to identify vehicle movements road by road in cities, specifically Barcelona. For this, I'm going to use high resolution satellite imagery, which has a 30 centimetre spatial resolution and daily temporal resolution. So a quirk of these satellites that I'm going to exploit to estimate vehicle speed is that there is a ½ a second gap between the data capture of the 8 spectral bands on the satellite. So to show you visually the quirk of this satellite, if you look at the image on the left, you can see that there are 2 coloured blobs at either end of the vehicles. And these blobs are showing the shift, or rather the movement of the vehicle over that ½ a second gap between the spectral band capture. So I'll be using this shift in location to estimate vehicle speed. On the right, I've shown an image of a subset of my training data set in Barcelona, which I manually created in QGIS. I split the vehicles into different movement types and 3 different vehicle sizes. And the yellow boxes show you all the identified vehicles in the scene. So I'm using the training data set to train a deep learning object detection model to identify the vehicles in satellite imagery. And as you can see from the table displaying the number of manual counts in the training data set across different movement types and vehicle sizes, the majority of the vehicles in cities, specifically here Barcelona, are parked. And as the aim of my project is to use the traffic data and air pollution modelling, we're mainly interested in those vehicles that are moving. So I'm incorporating movement information of the objects in my training data set to hopefully get the model to learn what a moving vehicle looks like. So to conclude, I'm currently training the model and using lots of different computer vision and remote sensing techniques to improve the accuracy of vehicle detections. And that's it. Thank you for listening.
Thank you very much, Annalisa. Really amazing. And finally, Aina Roca Barceló from Imperial College of London.
Yeah, thank you. Let me just share my screen. Okay, can you see my screen now? I guess so. Okay, then I am just gonna start. Thank you so much. So temperatures are expected to continue to change with climate change. So retrospective assessment of human and social adaptation to these changes is essential so that we can anticipate what are the needs and costs that are associated to these future changes. Here, I'm going to be exploring whether the population in Sao Paulo, which is a megacity in Brazil, which you can see here in the map, have adapted to temperature changes over the last 2 decades. And I'm going to be focusing mainly on the elderly and females. So we obtain temperature recordings from the University of Sao Paulo Meteorological Station, which you can see on the top left. It's considered a reference station and has been repeatedly used for temperature mortality studies previously. We took hourly recordings and we averaged them to obtain daily averages of temperature, which you can see plotted on the top right plot. Mortality records for non-external causes of deaths were obtained from the Ministry of Health in Brazil, and you can see them plotted on the bottom right. Here, it is important to note there is a strong seasonality in the death counts. We used a distributed lag nonlinear model, which allowed us to define both the behavior of the association on the lag and the predictor dimension. And so it allowed us to capture both the delayed and the nonlinear effects of temperature on mortality, respectively. I adjusted all the models for holiday, day of the week, seasonality, and long-term trends, the latter using a natural cubic spline with 10 degrees of freedom, which you can see fitted here in the plot with a blue line on the bottom right. The best model, the best fitting model was selected using QAC criteria. And for the purpose of evaluating adaptation, we stratified the analysis in 2 10-year periods from 2000 to 2010, and then from 2008 to 2018. Here, I show only the results for gender and age stratify analysis on your left and on your right, respectively. These plots show the relative risk on your y-axis associated to different temperatures, which you can see on the x-axis. And I plotted in bright pink the first period as opposed to the purple, which is the second period. And I shaded the 95 confidence intervals with the same color. I've also indicated with a solid vertical line of the same color the minimum mortality temperature, which is the temperature at which we register the lowest mortality risk and that we used as a reference to calculate the relative risks. And I will refer to these threshold temperature as the MMT from now on. So basically, if our hypothesis of adaptation is correct, we would expect either of these things or both to happen. On the one hand, the MMT to be higher during the second period, and or the slope of the relative risk to flatten over the second period. For all the groups that I present here, the MMT in the second period is higher, and that's especially true for females, which can be indicative of adaptation to higher temperatures. If we now focus on the slope and we look at the second period, for females, we can see that the second period during cold temperatures is slightly lower, and it is actually higher for hot temperatures. For males, however, the slope remains mainly unchanged for cold temperatures, and only a slight reduction can be observed for high temperatures. If we now look at the age differences and we focus on the eldest group on the bottom right, we can see that the risk remains virtually unchanged over the whole temperature spectrum. Whereas for the population less than 60 years old, we can see that there's a remarkable flattening of the relative curve for both high and low temperatures. So in conclusion, these results suggest that there is a slight adaptation in Sao Paulo specifically. And now at the moment, we're assessing different vulnerable groups in addition to the ones presented here. Thank you so much.
Thank you very much. So again, a big congratulations to all the 6 of you for the great talk you gave, for reaching the finals and for competing for the award that will be announced in more or less half an hour. Congratulations. So since we are in perfect time, even a little ahead of time, if there is any, let's say, doubt how to vote, etc., I think our technical chairs can share a slide on how to vote. And then we will move to the next session.
All right, so the vote's going to go live in about 1 second. Here we go. I think you all know the drill by now, but just to make sure, it's in the app. So on the app, you go to Vote for Speed Talks. On the web version, it's under Surveys. This is not applicable because you'll only see one, and that's the final. And same as before, 1 to 5, 1 being room for improvement, 5 being could not be more amazing, and you can't pause once you've started. So if you're in the app version, if you miss someone's talk, by the way, please just give this, I'm sure it was great rating. And in the app version, you'll see this confirmation screen when you're done. So app voting should now be live. I'll confirm that. And you guys have exactly 5 minutes now to submit your votes. Then we're going to close voting because we need to tally the votes, and we'll be announcing that soon. So congratulations, Speed Talkers. It's already an achievement just to be in the finals. This was wonderful. I really enjoyed it. And we'll see you soon when we announce our winner and runner-up. So I hand back to you now, Massimo.
Thank you very much. Great. So, okay, yes. I'm now glad to invite Mark Neuheisen, research professor at ISEE Global, but most importantly, president of the ISEE, who was very kind to participate here and bring to us the greetings from the society. Please, Mark.
Muted. Welcome everyone, and I've been given a few minutes to say a few words about the society. You heard before Michelle already, who introduced the society and also explained some of the benefits of joining the society. And I advise you all to become a member, and particularly become an active member for the society, because it's a great club to be with, and we tend to have very good fun. So actually, I would like to use these few minutes just to thank the local organizer and also the scientific committee for putting this great conference together. I've greatly enjoyed all the talks that I've seen. Amazing work that is ongoing, great quality, and the conference very well organized, very interesting. So it's been a great day, great couple of days. In a way, it's a shame that we couldn't be there in person. That's the only thing what I've missed, actually, to meet you all in person. But hopefully next year or the year after, probably more likely next year, when we also have the main conference in Athens. I hope that all of you are also coming to the Athens conference. I mean, it's not so far away, and it will be also a great conference. I hope that you keep going on with your work. I mean, it's always a nice milestone to see what is being, what you're doing by presenting the work. At the same time, I mean, I've said for many of you, you're halfway through your work, or You've got new work plans, so I'm hoping that you can continue with this work as well. As a society, I believe we're doing some very important work that's also quite relevant. I mean, I know it's gone a bit off the agenda with COVID, but I'm sure it will be back soon, and we've seen some COVID-related work as well. I will leave it for this because I know it's been a very intensive 2 days. I wish you all a very nice weekend and keep up all the good work. Thank you so much.
Thank you, Mark. Thank you for popping in. And yes, I miss also meeting people in live, but it will come back someday.
Very soon, I'm sure.
Okay, now it's the time of the awards. So I really wanted to thank the awards subcommittee. They did a great job. I really thank them. And now I leave the floor to you.
Thank you, Benedict. Let me share. You can all see the screen. Thank you. My congratulations also to both conference chairs and the local organizing committee. It's a consensus that has been a great success, this virtual conference in these weird times. And it was our job was hard because there were a lot of very interesting work that has been presented and very high quality research. The committee was myself, Evi Samoli from the University of Athens, Sarah Kress from Leibniz Research Institute in Düsseldorf, and Andrea Daleka from the University of Ostrava in the Czech Republic. And just to go everybody through the rationale of our choices for the best award for the oral presentations. Each abstract was rated for quality, originality, and impact. Our initial assessment was based on the evaluation of the abstracts from the external reviewers as part from the scientific committee job. And in case of there were ties in the highest ranked abstracts, all of these abstracts were reevaluated by the three of us. Regarding the awards for the best tweezer, our rationale was quite different. We decided that our choice will be based on a combination between a nice aesthetical result that will convey a clear message that will be clear regarding the conclusion of the research being presented. And we thought to award people that have worked a bit to edit their picture, their tweezer, and not just have some picture copied from a Google search or something like this. So before we announce, we open the envelopes with the winners. Let me introduce that the first prize for each category will be registration that the ISEE Council has graciously offered to cover the virtual conference registration for the first award in each of the categories. And then there are runner-ups. We have a second and third awardee for best abstract and the best tweezer, that they will get an excellent treat by regular mail with biscuits and renowned Swiss chocolate. So I put now the floor to Sarah to present the final awards.
Yeah, thank you. And we want to thank all of the candidates for their work, and people had put a lot of work on the abstract, and they cover a wide range of topics that we believe are guaranteed to make a successful conference. So we got very delightful insights into various interesting topics, and it was a really hard decision. So we hope that all of you get the same impressions after announcing our winners. So the first prize goes to, and it was a surprise for the winners too, so I hope you are here, Maciej van der Drijs from the ISEE Global in Spain, for his abstract on prenatal exposure to non-persistent chemical mixtures and offspring IQ and emotional and behavioral symptoms. Thank you for your work, Maciej, and congratulations. Okay.
Sarah, can you spotlight? Ah, he's here.
Yeah. Okay. Congratulations.
Sorry.
Right. Thank you very much.
Yeah. Okay. Yeah.
It's
So.
really great.
Okay, then we come to the next prizes. It's Alicia Ebeyen, again from the ISEE Global in Spain, for her abstract on early life exposure to urban environment and lung function asthma in school-aged children, the Generation R study. Alicia, congratulations.
Thank you.
And the third prize goes to Matteo Scortichini, working at the Department of Epidemiology of the Regional Health Service, Lazio in Italy, for his abstract on a countrywide small area analysis on temperature, mortality and risk, and geographical variation in vulnerability. So thank you and congratulations.
Thank you. Thank you very much.
Yeah, so thank you again for all the winners and all the candidates for their excellent work.
So should we move to the Tweezers Awards?
Yes, please. Andrea?
Yes, good afternoon, everybody. It is my great pleasure to announce the 3 best tweezers of the ISEE conference. And to be honest, it was very challenging for us to choosing the best tweezers because when we were selecting, it was evident that the presenters put a lot of effort to their creativity. But as Evie mentioned, we focused mostly on the combination of the creativity and strong and clear message of the tweezers. So now I would like to announce the first place of the tweezers, and it goes to Lea Metre from ICE Global. With her very amazing tweezers and very nice expression of the environmental exposures to a little child. And her speed talks or oral speaking relates to early life environmental exposures induce persistent changes in gut bacteria composition of 7-year-old children. So congratulations, Lea.
Unfortunately, Leah is not here in this session now.
Okay.
But I'm sure she'll be delighted when she gets the email.
Okay, so I will move on to the second and the third place of the our tweezers, and
I'm sorry.
and the second place goes to Yijin Ma from the Yale School of Public Health with her presentation related to role of meteorological factors in the transmission of COVID-19 in the United States. So congratulations, Yijin, for a very great tweezer.
Thank you.
And the third place goes to Emeline Legy from French INSERM, and her presentation related to estimating urban exposures to airborne metals using MOS biomonitoring. And also congratulations a lot for your very nice and original image of your tweezers.
Thank you so much. It's a pleasure.
Yes, now I think that I will take the floor back to Danielle, and maybe she will let us who is the winner of the speed talks.
I will do that. So we are, so 85 people voted. That's a lot. That's and well, first, congratulations to all the previous winners. Very well done. And also, so I am happy to give the award to the speed talk. So it will be only 2 here. And the second place is Carly Goodman.
Thank you very much.
And the first place is for Alexandra Burgel. Congratulations.
Thank you so much.
So, popular vote. Very well done. Okay, so thank you. We are almost ready to wrap up. Just before giving the floor to Danielle for final words, I would like to announce that I am very happy to share with you that we are hosting in Rennes the next ISEE Young Conference. So we still don't know the dates, etc. We will come with further details soon. So keep tuned, and we hope to see you there. And now, Danielle, the floor is yours.
Great, thanks.
Wrap up the conference.
Well, I'm happy if you stay here with me in spotlight. Oh, you disappeared. That's okay. Congratulations to everyone. That was really nice. I can put her back. Yeah, come on, Benedict, stay up here with me.
It's so lonely there in the spotlight, isn't it?
Yes.
You know, because the first thing we were going to do was to announce the leaderboard in the app, right? And I think this has been something you've been keeping your eye on throughout the conference, Benedict. Yes. You know, I was the winner there for a little while during the testing phase, and then the conference kicked off, and you seem to really take advantage of our app, which we're very grateful for. You still are in the lead, but I'm afraid to tell you, you're not eligible.
Okay.
No, so, and in fact, it has been a fierce bit of competition going on. And I can now say, because everyone is probably looking at their app with me, that our winner here is Meltem from Swiss TPH. She just took over the lead, I think, sometime during this plenary. So congratulations, Meltem. There's also a prize coming your way. So with that,
yay.
Great. So I have the pleasure to do the thanking at the end here. Thanks, first of all, to the ISEE Council, you know, to Mark and Michelle and everybody else on that council for really the support and interest in ISEE Young over the years, and for helping us. And of course, for sponsoring those first place awards. So that also includes the Speed Talk Award, that first place, the conference registration. Congratulations to our 3 winners. And thank you to the ISEE for making that donation to us. Really also want to thank the Europe chapter. So you guys all know who you are, and the SOC run by Benedict and Massimo. You guys did a fantastic job putting together the program, putting together the team of volunteers that have contributed to the SOC. So everybody that were our abstract reviewers, all of our chairs and co-chairs over these last 2 days, it's been fantastic. And then for the committees, the awards committee that we just saw, thank you very, very much. I know you had a lot of work to do. And also the networking committee. There were some fantastic events. I went to all of them, and I was very impressed, and thanks for that. So also, of course, there's people here at Swiss TPH that we wouldn't have been able to do this without them. These are the departments of communications and finance that were behind the scenes helping us in different ways. And also our very own Department of Epidemiology and Public Health. And there in particular, Nicole for really saying, yes, do this, you know, have fun and go for it. And that's Nicole Probst. And Martin Roosley, he's really dedicated a lot of his staff time and also his interest in helping us promote the conference and tweeting for us during these past few days in advance. So thanks for that. But really the biggest thanks goes to the rest of the local organizing committee. Marloes Eftens, she put in so much time and effort. Thank you very much. And Anna Vicero, joining us a little bit remotely, but still part of our local group. We really appreciate everything that you've done as well. But, you know, really, our 2 stars are, I mean, this would not have been possible without the dedication, the time, energy, enthusiasm from our very 2 special PhD students who you've all met by now many times, Apolline and Alexandra. We really, oh, this is great. Bring them on the screen. Thanks a lot. I know everybody's clapping for you. And I just wanted to say that, you know, while Marloes and we share an office, while we were having discussions over the past year, earlier in the COVID crisis, we don't have Marloes on screen. If we can find her, I'll talk a bit slower.
She was hiding, but I've
hiding. Okay.
Sorry,
Guys,
I
you
had
can
my
unmute
camera
now
turned
as well.
off.
Okay,
Okay, there you are. I'm gonna find you now.
I'll let you find Marloes. Well, I tell a little story that, you know, we always discuss things, and we were thinking about whether we were going to be forced online. I'm pretty sure that Apolline and Alexandra were already figuring out brilliant ways to make this, you know, online conference happen. Sorry, I don't think I said that quite right. While we were still wondering what was going to happen, they were planning and really, you know, making Zoom backgrounds and networking events. I'm sure they started it well before we had even thought that that was the direction that we were going in. So really, you know, last but not least, I want to express just enormous gratitude and congratulations to this technical team. Again, these are people that you have met throughout the conference. And I mean, they really brought this conference alive for me, for you. I think everybody agrees with this. So Sarah, I know you like to hide back there, but Sarah Thiele, your outstanding leadership, brilliant organization, and communication skills have just made this virtual conference, the ISEE Young, such a success, in my opinion. And I should also say that along with Apolline and Alexandra, the three of you really know how to bring together a superb team. There are those volunteers, our student volunteers, that have really worked tirelessly these last 2 days and days in advance, training, preparing for this, making sure that we could just have everything run as smoothly as it did. So yes, let's bring them on screen. If it, or
I'm running out of spotlight, so I'm taking some of you off, okay?
Okay. You can make me go away too.
No.
Yes, so I will like to, I mean, it's my pleasure to introduce the whole team that we can find to bring here. In Munster Room, along with Sarah, we had, shall I go?
I think some of them have they've been working so hard. They've been here
disappeared.
so long, and they left just a bit early. A lot of them were here, but Alonzo and Brian are missing.
Okay, well, anyway, we've had, yes, Alonzo, and Brian actually was supporting us all the way from Kenya. And so that was also really fantastic. Thank you for that. And also in the Munster Room, we had Sophie Baruth, who's here on screen with us. And she's a trainee that just joined us. Oh, hello.
Oh, no, I'm sorry.
It's okay. And thank you for just jumping in with 2 feet and helping us with the conference. So we also had our volunteers in Rat House, which what are we in now? We had Lydia, Fabienne, Tafadzva. Thank you so much. I'm making it very complicated here for Sarah.
I mean, it's just the home stretch. We're almost there.
Exactly. And we also had our Spallantor volunteers. So we had Louise, again, joining us remotely, Nicholas here on screen, and Brady, I think you said he was probably off having a rest as well. Thank you so much. Have I captured everybody? Have I said congratulations to you all? Listen, we're so grateful. Thank you.
I'm sure there's someone that we may have forgotten in this, and apologies, because there were just so many hands on deck to make this a big success. So if we forgot you, please don't take it personally. We really appreciate everything everyone has done to make this possible.
Exactly. So round of applause for all of you. And Sarah, if you don't mind, if I could come back in the spotlight just with Benedict for a moment.
All right. Good.
Hi, Benedict. I wanted you back here. So I want to, well, we're going to close the conference together. So that's what's coming next. But first I want to say, I'm really excited that you're going to take on the next ISEE Young, whenever that may be. And so here I am handing over the torch to you. And I look forward to, supporting you as you've supported us in the next ISEE Young conference.
Thank you very much. And thank you all again.
And we'll just say
you very soon. Bye.
Bye-bye.
Thank you all.